amylase. Amylase is an enzyme that breaks down starch. We're going to try and find out what its optimum pH is today. So optimum means the best pH that it'll work at. So if you come in, I'll show you the equipment that we're using. So first of all, we need 2 centimetres cubed of amylase. We're going to use a syringe to do this, to be really accurate. So we add that to a test tube. And we're going to follow that up by adding our pH of choice. Now we're going to use pH 2. So you add 1 centimetre cubed of pH 2 in a different syringe and add that to your mixture. Okay. The next thing that you're going to do is you're going to allow them to mix, and you're going to add a drop of iodine to each dimple in a dimple tray. Now, we're using iodine because iodine turns black when starch is present. So, as the starch breaks down, the colour should change from black back to a brown colour. Okay. We're going to take our mixture from before and we're going to add 2 centimetres cubed of starch. As soon as I've added 2 centimetres cubed, I'm going to start my stopwatch because the amylase will have started to work and we'll be starting to break down the starch into sugar. I'm going to leave the stopwatch there. Next thing that we're going to do is every 20 seconds we are going to add some of our mixture to a dropper, to a dimple. As you can see, it's turned black because the starch is present. We're going to add another drop to the next dimple in another 20 seconds. Over time, what will happen is the colour will change from blue or black to brown again. Okay, welcome back. So, we left this for a little while. We've been doing it for 2 minutes and 20 seconds. And as you can see, as the time has gone on from 20 to 40 to 60 to 80 to 100, 120, 140 seconds, the colour has changed until there is no more blue. That means all of the starch has been broken down by the amylase. So we're going to record this time and we're going to repeat the experiment with several different pHs. As you can see, we've got pH 2 here, but we're going to use some others all the way up to pH 14 if you want. Now, we're going to plot our results on a graph, plotting time against pH. And as you can see, you will get this sort of curve. Okay? Now, as you increase the pH, the time will decrease because it will take less time for the enzyme to break down the starch. This point here at the bottom is the quickest time. This is the optimum pH. As you can see on our graph, it is 7. For different enzymes, it's different. That means all the starch was broken down very quickly. After pH 7, you can see that the line starts to increase again and time starts to increase. This is because your enzyme has denatured. That means that the active site has changed and will no longer be able to break down the starch molecules. In an exam question, you may be given, instead of a time graph, a rate graph, which will be the opposite way around. But the same idea still applies. So your optimum pH is still going to be the one where you get either a peak or a trough. Okay, thank you.